Okay, last talk before the panel. Improving computational models of human antibodies by Bryan Briney, who's an associate professor at Scripps.
All right. So yes, I'm the last speaker before we go to, let's see. Oops, that's the go-ahead slide, I guess, huh? All right, so I'm the last speaker before we go to the panel and then dinner. So that's bad because I'm the last thing holding you from dinner. But on the upside, I got to follow a bunch of amazing speakers that did a really, really good job of presenting kind of the challenges we're facing in this kind of AI computational modeling arena, specifically as it relates to antibodies. So my group does a lot of antibody discovery, and our primary technique is using this antigen barcoding approach. This is now commercially available through 10x. It's called BMAB, which Victor very nicely called supercharged LibreSeq, which I appreciate. I'll put that on the back of the book, you know, like Victor Grief. Yeah. So the general approach is we take protein antigens and conjugate them to a DNA barcode, which is unique per antigen, as well as a fluorophore. We can sort antigen-specific B cells, and then use a single-cell platform like 10x Genomics. And the B cells will then drag these barcoded droplets into our barcoded antigens into the droplets with them, and we can sequence the barcode information along with all the other single-cell omics stuff we can get. So transcriptional profiles, paired antibody sequences, all of those things. In a single example of how this would work, this is also in collaboration with 10x. This is antibodies from a single donor, who was infected relatively early in the SARS-2 pandemic, and we were able to recover a total of just under 3,000 SARS-CoV-2 specific antibodies, of which we recombinantly made 240 or 239, and you can see the pretty extensive variability in terms of their binding specificities and cross-reactivity patterns. Now, the downside, of course, is that we were only able to make less than 10% of these and really do comprehensive characterization on a very small number of these antibodies. So how are we sure that we are looking at the best antibodies and that the best ones aren't in those other 90% that we weren't able to actually test? So there's a lot of ways to get better at this. We're clearly not great at it because only about half of the antibodies we're neutralizing, right? So we're not there yet. So there's a lot of ways to do this. So Brandon was talking earlier about ways to make functional screening far more high throughput and less expensive, which is definitely a path forward, which is very important. And I think we're taking a slightly different approach, but also complementary, which is that we need to get better antibody models. That's our kind of pitch. So we can recover many more antibodies now than we can test. So how do we make sure that we're not losing the best stuff, that we're not just overlooking the most interesting biology or the most appropriate or interesting antibodies? And so the reason this is so appealing is because antibody sequences encode all of the information you need to get structure and function. There's no extra information that gets added later that tells the antibody how to function or how to fold into a structure. It's there in the primary sequence. We just need tools that let us extract that information in a usable form. So there's lots of models that could potentially be made. Structure models, we just heard to talk about those. Functional predictive models, even things like vaccine elicitability, which I don't even know if is a word, but you get the idea. Is this antibody able to be elicited by a vaccine in a large fraction of the population? And the really cool part about this from kind of the academic side of things is these don't have to be perfect to be useful, right? So if you wanted to bin antibodies by specificity, you don't necessarily need antibody epitope mapping at atomic resolution. If you got the equivalent of a negative stain EM, where you can see the footprint of the antibody on the antigen, that's good enough for looking for antibodies that compete for binding to the same epitope region. So we can use very well models that don't necessarily need to be perfect. So we used large language models to do this, and initially you might think, well, what does a language model have to do with proteins? At a high level, they're actually pretty similar, right? So text is just a sequence of words, right? And proteins and antibodies as well, it's essentially a sequence of words. We just have a small vocabulary of 20 words. So just like antibodies and proteins, the order and the context that words appear in a sentence also have a big impact on its meaning, right? Same thing with proteins. If you change amino acids around or change the context in which they occur, you can have pretty large effects on the function and the structure of the protein. So the current state of the art in this area is a model architecture called transformers. Jane just mentioned those briefly. So unlike other neural networks, it doesn't use convolution, doesn't use recurrence. It uses a concept called attention. And so one way to think about that is you would look at going through every position in your sequence. From that position, you would look at every other position in the sequence to see how it relates to the position you're in, right? And the process of training this model, a transformer model, is the process of assigning weights to each one of those attention relationships. So you want to figure out how much attention should you be paying to your surrounding residues. In a protein context, think about being a cysteine. If you're at a cysteine, you should be paying a lot of attention to the other cysteines in the protein with you, right? That's the idea. So it allows your model to learn these pairwise relationships across the entire input sequence. So these models, unfortunately, take massive amounts of data to train and massive amounts to compute to train as well. So this is the data used to train a text model called Llama, which was released. It's actually only released maybe 4 or 5 months ago, and it's already not the state of the art. There's already a Llama 2 from Meta. And so they used about 5 terabytes of training data to do this. So that's a lot. If you printed it out on pages of paper, it would be about 150 miles high, or I guess I should have changed that to kilometers, so like 250 kilometers tall, right? Which is pretty tall. That's a lot of paper. And much, much more data than we have access to in terms of antibodies. So the people at Meta who trained this model were lucky in one sense, is that there is the internet of text, right? You can download massive amounts of text that's essentially freely available. And we don't quite have the equivalent of that for proteins or antibodies. So one aspect of these models that can be especially appealing, though, is that they can be function as what are called foundation models. So you can train one massive model on proteins or antibodies, and then use that knowledge for a variety of tasks by a process called fine-tuning, which I'll talk about later. So how can we make antibody language models better? First off, why should we do an antibody-specific language model? Why not just use a protein model? It turns out that antibody language models can perform better than protein language models on antibody-specific tasks. This has been shown for several different kinds of tasks, clustering things by identity or antibody characteristics. Antibody language models are better. They're better at performing structure prediction than even state-of-the-art models. So there is a benefit to training an antibody-specific model. One limitation of the existing models, however, that we noted was that all of them have been trained exclusively on unpaired sequences. So everyone here knows what antibodies are, I'm assuming. So the antigen recognition site includes both the heavy and the light chain. And those residues have to, the paratope has to act in concert across the heavy and the light chain. So if you're training with only unpaired data, you're unable to learn any features that span both heavy and light chains. And we thought that was a pretty important aspect of how antibodies function and should be included in these kinds of models. So we trained 2 models, baseline antibody language models called BOM. And an interesting story, so this is a preprint already. And a couple of days after we posted that, another group published another antibody language model also called BOM. So heads up, there's 2 of them out there already. So there's only so many words that have LM in it, so I think we've gotten close to using them all up already. So we trained 2 models, all of them using what is the largest publicly available data set of paired antibodies. It's about 1.5 million antibodies. So for perspective, the unpaired models that exist that are antibody-specific are all trained on tens to hundreds of millions of sequences. So we're orders of magnitude less than that in terms of training data. So it's unlikely that we're going to make the ultimate best model out there. We're just too data limited. But so our primary objective now was how much does paired antibodies improve the ability of language models to function? So to do that, we trained 2 models using the same data. The same 1.5 million paired sequences, one in which we trained with paired data and one in which we broke the pairing information and just trained the model on single sequences one at a time. And so what we transformer models are usually trained using a process called masked language modeling. And the way this process works is you give an input sequence. In this case, it's a concatenated heavy and light chain. You randomly select positions across the input for masking. You mask those residues, and then ask the model to predict which residues should go in those masked positions. Now, if the model is able to do that accurately, it has to learn a lot about the biophysical properties of amino acids, the context in which certain amino acids are most likely to occur, and also which amino acids are essentially replaceable with each other, right? That would mean that they're similar biophysically in the sense that you can swap one out and not disturb the protein structure or function. So we took our data set and we split this into 3 subsets: training, testing, and evaluation. That's going to be important in just a minute. And the reason we do that is you can train your data set on the training data, and you can test your data set once it's been test your model once it's been fully trained using a data set that it's never seen before. That's important because we don't want a model that memorizes things. We want a model that learns generalizable features about how antibodies work, right? So if you want to learn math, you don't just have flashcards and then look at the same flashcards again on your test. You want to learn a concept that allows you to add any 2 numbers that you see in the future, right? That's the kind of idea we want to do here. So we had a fantastic introduction to this. Jane just talked about this, so I can actually breeze through this a bit. Once we've trained the model, we can introspect a little bit and find out what the model knows. So if we take the output embeddings of the final layer and we cluster them using UMAP, we can see that, so on the top 2 plots are the paired model. The bottom 2 plots are the unpaired model. The left is coloring the clusters by V gene, and the right is coloring by somatic mutation. And just like we saw with Antibody, the model separates things primarily by mutation, which would be memory versus naive B cells, and secondarily by V gene. So you can see that there's 2 the green cluster is V3. You can see that there's 2 V3 clusters, primary clusters, one that's germline sequences and one that's mutated sequences. So we're dividing first by mutation, second by V gene. And so we didn't see much improvement between the unpaired model and the paired model. It looks pretty similar. Where we did see a big difference, though, is when we look at the light chain. So in the paired model, we see something very similar to the heavy chain. We see the sequences clustering primarily by somatic mutation, and then secondarily by their V gene use. There's a lot more V genes because this is kappa and lambda chains altogether, so we have a lot more clusters. But in the unpaired model, we see something totally different. We see some clustering by somatic mutation, but the germline sequences are diffuse and not well clustered at all. Which, when I saw this, I thought that's strange, right? So by including the heavy chain, you improve how well the model understands the light chain. All you have to do is give it the right paired heavy chain, and somehow it understands the light chain better. So that's a really interesting concept, but it actually fits with what's known immunologically. So there's a lot of data, emerging data and data from quite a while ago, that suggests that pairing is not random. So similar heavy chains tend to be paired with light chains that are also similar. But that's not asymmetric. Light chains are promiscuous. You can have the same light chain paired with a bunch of different heavy chains. So that fits with exactly what we see here. The heavy chain can better inform what the light chain looks like, but the light chain shouldn't necessarily inform what the heavy chain looks like because that same light chain can promiscuously pair with a lot of different heavy chains. So it does seem like we're learning these cross-chain features, right? One chain is informing what the model knows about the second chain. So how deep can we go, right? So we know that the model understands somatic mutation, and we also know that it can do cross-chain learning. So can we kind of supercharge that even one step further? So what we did was we took 15,000 mutated test sequences. Again, these are sequences the model's never seen before. We held them out during training. And what we can do is the sequences that we used had to have at least 3 mutations in each of the heavy and light chain. What we did was we masked all of the mutations in the heavy chain and then paired that heavy chain with either the naive or the native mutated light chain or a light chain where we reverted all of the mutations back to germline. Right? So what we would expect is if the model really kind of understands how these things work, the model should be able to recognize that a mutated light chain implies a mutated heavy chain, and an unmutated light chain implies an unmutated heavy chain. And that's exactly what we saw. when paired with a mutated light chain, the model is much more likely to assume or predict a mutated residue at that position, at each of the mutated positions. And so to make sure that we weren't seeing some sort of memorization, while the model is more likely to predict the bottom plot down here, while the model is more likely to predict the correct mutated residue, it's also much more likely to predict any non-germline residue. So this isn't a case where the model just memorized, well, this light chain goes with this mutation. It realizes that a mutated light chain goes with a mutated heavy chain. And so if we do the converse, or the complementary experiment where we mask the light chain and then pair the light chain with either the mutated or the unmutated heavy chain, we see the exact same phenomenon, right? So being paired with a mutated heavy chain makes the model much more likely to predict or assume a mutated partner chain. So I've mentioned a few times that we don't have enough paired data, right? So we can't necessarily make a state-of-the-art language model that can do things like protein prediction or something like that, structure prediction. But what can we do? Well, there's one way to kind of leverage the data we have along with other data that already exist. It's a process called transfer learning, and it's a way of taking a big model and somehow taking what it knows and applying it in a slightly new way, in a different direction. So one way to do that is this. It's called ESM-fold, right? So this was published by Meta also, and it does something similar to AlphaFold. It's not as accurate, it's not as good, but instead of using multiple sequence alignments as part of the input, it uses a protein language model called ESM2. And I think it's pretty widely agreed that this is the state-of-the-art protein language model. So remember, we have this matrix of numbers, which is the output embedding of the model. It's a huge list of numbers that includes all of the information the model knows about an input sequence. So rather than just giving the structure prediction portion of the model the sequence directly, you also give it these output embeddings that contain information about the sequence that's kind of in model language. So you have this protein language model on the left, which is trained, and then frozen, and then you put this structure prediction head on top of it that uses the information learned by the language model to now be presumably more accurate at structure prediction. So there's two ways that I'm going to talk about here about how to train, to fine-tune models. There's lots of ways to do it. This is just the two that we've used. So I'm going to describe them here. One of them is the one on the left in which you unfreeze the weights of the source model, usually a very large model, and essentially just keep training it with new data, right? So you would assume the model would then retain the information it used to have and then update itself to include the new information you've given it. The second on the right is essentially what ESM-fold did. You take the base ESM model, you don't change the weights at all, and you use the output of that model to train a second model on top that can do some sort of fine-tuned task. In this case, of ESM2 was structure prediction. So we fine-tuned ESM2 using the left-hand approach. So we have 1.5 million paired antibodies. We unfroze the weights of ESM2 and continued training with these antibody sequences to try to teach it additional information about antibodies and not just general protein understanding. So if you pass the model, the unfine-tuned, so the base ESM model, if you give that model an antibody sequence, in this particular case, it's a clinically approved COVID-19 antibody called Mesavibart. We did this for a bunch of antibodies, and this isn't like representative in the air quote sense. It really is. They all look pretty much like this. And so it seems like this is an attention map, a cross-chain attention map. So we're looking at every position in the heavy chain and seeing how much attention is paid to every position in the light chain, right? So only cross-attention. What we see is that the model is actually paying pretty close attention to cysteines, which is interesting, and again, appropriate for a language model, or for a protein language model. It's also paying close attention to the area right at the end of the heavy chain and the beginning of the light chain. And why would that be the case? Well, when we give the protein sequence as input, it's the heavy chain concatenated to the light chain. So it's just paying attention to residues that are actually consecutive in the input sequence, right? So it's just looking at things that are right next to itself, right? If we look at the fine-tuned model, it's a totally different story. So now what you can see is the cross-chain attention in the heavy chain is focused in the CDR3s of the heavy chain, and those CDRs are attending to the CDRs of the light chain as well. So if you look at the heavy chain CDR3, this may be not working. The heavy chain CDR3, if you look at the attention going across the light chain, you can see that most of the attention is in CDRs 1, 2, and 3 of the light chain, which, again, correspond to the antigen recognition site of the antibody, which suggests the model is learning something immunologically relevant about antibodies, but they're also structurally proximal as well. So it's potentially learning something about the structure of the protein as well. The pairwise residues that it's paying most attention to are the ones that are structurally proximal and most likely to be related to function. So that's very interesting. So now can we do something else with this? It's a little more practical, right? So we wanted to try to do a fairly simple, fairly low-bar experiment where we tried to classify antibodies by specificity. This is a binary classification. So we took from COVABDAB, which is a database of coronavirus antibody sequences. We took all of those sequences, we clustered them, and again, we wanted to cluster them so we make sure that there's not the same thing in your training and your test data. We don't want to memorize, we want to make sure we're learning something fundamental here. So we clustered them so to make sure that the test and train data sets are distinct, and then we're left with about 9,000 coronavirus-specific antibodies. We did the same thing with control sequences from just healthy donors, memory B cells from healthy donors, and a matched size data set. And then we trained a sequence classification model on top of the fine-tuned ESM model. We did the same thing for the base ESM model as well. So we have a base ESM model with a classification head, and we have our fine-tuned model with a classification head as well. And what we can see is that the fine-tuned ESM model does do better. It's not overwhelmingly better. We're in the 81, 82% range in terms of accuracy. For the base ESM model, it's more like 77%, which is a decent difference, but not incredible. Now remember, ESM2, the base model, was trained with 500 million protein sequences, and we fine-tuned it with 1.5 million. So that may be part of the reason. Fine-tuning with a much larger data set may produce a much better result. What we do see, though, is that during training, if you evaluate the model during the training process, we can see that the fine-tuned model learns much more rapidly, the classification head. So what does that mean? Well, more quickly means time, right, and compute, of course, but it also means in the number of sequences. So we had about 9,000 sequences, and we got to 80-ish%, right? But the fine-tuned model was already at 74% after just 1,000 sequences, whereas ESM2, the base model, was still at 59%. And just as a reminder, this is a binary classification, so 50% is guessing, right? If you have a 50-50 shot, you're getting it right 50% of the time. So 60% is a little better than guessing, but not incredible. So we see a substantial difference with a limited data set. So you can't imagine how this model can be, even if the top line number isn't necessarily dramatically better, we can actually fine-tune a much better model with much less data than you can using a base model. So if we dig a little bit deeper into what the model is learning and how it's learning, one of the things we notice is the outputs of the fine-tuning, the classification head, aren't like COV-specific or not, right? It gives you a probability per class. We have two classes, COV no and COV yes, right? And so we're going to get a probability for each. What we can see is that if the probability of the positive class ranges from 0 to 1, where the model is most certain, which is 0%, meaning 100% not COV-specific, or 1, which is 100% COV-specific, it's very accurate. Where it's not very good is right around that 0.5 marker, which is the model's like 51% COV-specific, 49% COV-specific. So this is actually very promising in the sense that the model's confidence does actually correlate with correctness or accuracy. And if we dig a little deeper into that, what we can see is as the model confidence increases, we see that the top plot is showing model confidence along the x-axis and accuracy across the y-axis. So at each one of those positions, that's looking at all sequences that are that level of confidence or higher, right? So you can see an almost linear increase with confidence versus accuracy. And again, if you look at the bottom, you can look at model confidence versus how many of the test sequences are that confidence level or higher. And that can help us set kind of this like almost volume tuning knob for how accurate you want the model to be. So my personal feeling is the sweet spot is somewhere between 0.8 and 0.9, meaning 80 or 90% confident. That means you're still looking at half to 2/3 of the data set is that confidence level or higher, and your accuracy in that 0.8 to 0.9 range is 90 to 95% in terms of binary classification, which I think is surprisingly high. So again, we were a little worried, is the model just kind of memorizing things? So even though we clustered, are there just does the model perform best on a test sequence when there's something very similar in the trained data set, right? Is it just learning similarity, not some sort of actual pattern with coronavirus specificity? And the answer seems to be no. So what we did here is we took every test sequence and found the most similar training data sequence in terms of identity. And what we can see is that the incorrect predictions and the correct predictions have a roughly similar distribution in terms of how similar the most similar training data set is. If this was not the case, if almost all the correct predictions were very high and all of the incorrect predictions were very low, it's probably not learning a general pattern of cove specificity. It's probably learning, hey, I saw one just like this. That's probably the match, right? And if we go even deeper into that, this is a plot showing every test data set and the model confidence across the x-axis. On the y-axis, the identity to the closest training data set sequence. And they're colored by whether the labels match. So labels match means the test and the train sequence are both the same specificity, right? So what we can see, what I thought was actually fascinating, I immediately went to the bottom right corner, right? And there's a dot down there that's about 70% similar to the train sequence, but they have the same label. Right? And the model is almost 100% confident. So you have these sequences that are dramatically different, and the model is extremely confident they have the same function in terms of classification. So again, it does appear that the model is learning some general patterns of specificity to coronaviruses and not necessarily just kind of finding very similar sequences and matching them up. So there's a bunch of stuff we can do next, but the most important thing, and we're kind of getting a little bit short on time, and I don't want to keep you any longer from dinner than we have to, but so there's a lot of ways we can improve these. Number one, bigger models. So bigger models require more data, but as a general rule, they tend to be more accurate, especially at the size we're talking about now. We're not into the kind of GPT level stuff. We're in the hundreds of millions of parameters, not the hundreds of billions of parameters. So the other possibility is, should we retrain ESM2 from scratch using all antibody sequences, right? Can we train a model using unpaired sequences and then fine-tune using paired sequences and get the best of both worlds? The vast data sets of unpaired antibodies, but then you also link in the pairing information we get from natively paired sequences. That's a possibility as well. Or would it even be better to do a mixture of both, where we have a mixture of protein sequences and antibody sequences roughly in an equal mix, so that the model learns things that are generally protein-related, but also has an enhanced focus on antibodies. So there's also a lot of ways we can improve the classifier. So a binary classifier in practice isn't that awesome, right? I don't think there's a bunch of people out here being like, I have this antibody, can you tell me if it's coronavirus specific or not? That's not a super broad use case, right? But number one, we should introspect into these models to find out what it's learning. What features allow this model to confidently describe an antibody as specific to a viral pathogen or not? The second, though, is moving beyond binary classification. So if we could do 50-way classification with the 50 most common respiratory viruses, now you're talking about something really interesting, right? I could give you an antibody and you could say, well, it's not RSV, it's not MPV, it's probably flu, right? Now you're talking about something that's actually more useful and generalizable to kind of a much more broader range of uses. And then also, does the model confidence correlate with goodness, I guess, I'm making up words all over the place, but if the model is more confident, does that mean the sequence is a better coronavirus antibody, right? So could we use that confidence as a proxy for engineering? If you take an antibody that's like a 65% coronavirus antibody, make a few mutations, and now it's a 90% antibody, does that mean it's actually higher affinity or more potent or better in some real way? Right? We still don't know that yet either. And again, there's other fine-tuning objectives. We can take this massive protein antibody language model and do things like structure prediction, epitope classification, and all of these other things. So again, and this has been mentioned several times before, not just by me, this entire thing is mostly data limiting, right? I think we do have model architectures that are sufficient to do these kinds of tasks. We just need more data, and the data has to be high-quality data as well. So briefly, some acknowledgments. So most of the work was done by two very talented people in my lab. So Sarah Burbach, who is a research programmer, did most of the AI stuff, and Jonathan was responsible for most of the COVID antibody stuff. And there's a rule in my lab where if you have a dog or a pet, you have to show a picture on the acknowledgement slide because they make our lives better and whatever else. So this is Charlie having a role at the park. And of course, we'd like to thank for the funding. And I think Samir mentioned this earlier. Data is very important, and having the data be openly available is very important. So all of the code we used is available. It's open source. You can download it and try it today. All of the models, the trained model weights, are all available. Our training data is all available. You can download it right now if you wanted to and start playing with it. So, and that QR code is for the GitHub, which has links to everything you want to get to. Thank you.